Hello, Valencia College and Dr. Saad. My name is Aiden Freihan. Today I'm going to be presenting to you DNA replication for Biology 1. So what is DNA replication? Well, DNA replication is taking one strand of DNA and splitting it into two. Our body does this every day in our cells, splitting and replicating DNA so that we can continue to grow and develop as humans. Other organisms do this as well, but today I'm only going to be presenting to you eukaryotic cells in humans. So in order to understand DNA replication, you have to understand what DNA is made of. So DNA is made up of antiparallel strands of both sugars and phosphates. And in between the antiparallel strands, we have nucleotides, which is basically genetic coding, to simplify it. So we have adenine, which stands for A, guanine, cytosine, and thymine that are all held together through peptide bonds. Hydrogen bonds. The phosphate and sugars are held together through peptide bonds. And so in order for DNA to get from this stage to this stage, there's a lot of work that needs to be done. So what is the first step? The first step, and excuse my drawings, they're not as good as Dr. Saad's in his videos. But in order to get from this to this, we need to first split the one strand of DNA into two separate ones. So that is the job of topoisomerase. It makes it a large kind of bubble, as you can see in this picture. And this line is called the origin of replication. And so that's where eventually it will split into two separate strands. And as it splits, it goes kind of like this, and it is unwound, and it is held together by these little proteins called single-strand binding proteins, and they run along both sides. And also, I forgot to mention that the antiparallel strands run from 5' to 3', and the other side goes from 3' to 5'. This means that the 5' end has the carbon facing upward, and this is a way to remember it, the 3' has the carbon facing downward, so it's running anti-parallel to one another. So to continue, topoisomerase expands it, and it starts to split into two separate strands through the enzyme called helicase. So helicase kind of runs through the hydrogen bonds and breaks them apart and splits them into two. And so these will kind of start to separate. And as it does that, we have a protein called DNA polymerase. So this DNA polymerase is actually very happy. He is going from the 5' to 3' direction, and he is easily able to duplicate this area, but not first without having a primer, like one of these. It would usually be maybe somewhere right here. And it would start here and just continue and make all of the nucleotides that pair appropriately. This DNA polymerase, on the other hand, is actually somewhat upset. Why is he upset? Well, remember how I said this one is running from the 5' to 3' direction, but this one cannot. It's actually going to 3' to 5' direction. So what has to happen is the primase, which makes the primer, puts one down. The DNA polymerase will come through and make a fragment. And then it will happen over and over and over. And those fragments are called Okazaki fragments. So it will come in, it will make the Okazaki fragments, it will come through, make multiple Okazaki fragments, and this DNA polymerase will easily be able to come through. But there are gaps in the middle, so how do you fix the gaps? Well, the DNA polymerase 1, which I didn't draw on this example, will come in and remove the primer from the DNA strand. And then a protein called ligase will come in and close the gap. It's a very minuscule and small gap, but it still needs to be closed. So in the end, all the gaps are closed, as you can see. And then at the very end comes your 2 strands of DNA. So thank you all. I hope this was very helpful in understanding the two strands of DNA from one strand and DNA replication. My name is Aiden Freihat, and I thank you for listening. And that was my presentation on DNA replication.